Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. I think we need to finish chapter 13 and then start chapter 7. 'We get to there or we further? Yeah, we're there. All right. All right. Before we begin, are there any questions? Let me open my screen. Share my screen. That's what I want to say. All right, should be seeing my screen now. There's not much up right now, but just let me get out of here. I guess I'll leave. Nah, I don't really need that, but we'll leave it. Got it open here somewhere. All right. Today is December 5th, and this will be the last class for microbiology. We're supposed to be covering chapter 7 today. We need to finish chapter 13, and then I'll get into chapter 7. I've got here a review. I'm going to post a review in the lab, hopefully right after class, where it'll be a review that I made in a previous term, and it will be a nice review of chapter 13, 14, 8, and the previous earlier chapters. It does not review chapter 7 because this review was made in the summer, and I don't cover chapter 7 in the summer. So I will try and do a review of chapter 7 after we cover chapter 7 today. Any questions about any of that? All right. Let's move to chapter 13, and hopefully we'll finish this quickly. I have that we had talked about latent and persistent viral infections, and then I don't know, the number of virions and the time course of those infections. So let's move on and talk about prions. Prion stands for proteinaceous infectious particles. And a prion is a protein that actually can infect a person and then cause disease. Prions can be inherited, meaning you can get it from your parents. They can be transmissible by ingestion of the prion, by transplant, like an organ transplant, bringing in the prion, and by surgical instruments. Prions cause a number of diseases, including spongiform encephalopathies. These are vacuoles in the brain in humans and other animals. Let me show you that now. Blow this slide up. So this is the appearance of a normal human brain. These are all prion infected brains, and you can see the brains sort of look like a sponge. There's holes in the brain, and that's what it makes the appearance of a sponge. And that's where the term encephalopathy comes from. Let me see. I think I want to end that there and go back to here. I mean, spongiform encephalopathy comes from because the encephalopathy takes the appearance of a sponge. In animals, the most common diseases are bovine spongiform encephalopathy, which is more commonly called mad cow disease. But also there's sheep scrapies, a disease European ranchers have been dealing with for centuries, and then cervid chronic wasting disease. In humans, we don't tend to call them spongiform encephalopathy. They're called either Creutzfeldt-Jakob disease, Gerstmann, Straussler, Schneckner syndrome, and I could be butchering that name. I'm not German. And then fatal familial insomnia, and then Kuru. All of these diseases are caused by prions. The Creutzfeldt-Jakob disease and Gerstmann, Straussler, Schneckner syndrome are diseases that are inherited in certain human families. We'll talk a little bit about Kuru. Kuru was a disease seen in the Fore tribe in Papua New Guinea. There's Papua New Guinea. And I blow this up a little bit. This was the region of the Fore tribe where the patient became unable to walk, loss of the ability to swallow or chew, drastic weight loss, and then what do you call that? Lethargy, leading eventually to death. How was it transmitted? In the Fore tribe, they had an unusual ritual in their funerals, which involved people cooking their dead relatives and then eating them.
Mm-mm. Oh my
Somebody's
gosh.
got your microphone on. Go ahead and turn that off. That was a very unusual ritual, funeral ritual, and people were eating the brains and the nervous tissue of their dead relatives, and that somebody in the Fore tribe came down with Kuru, and then it was spread by the funeral ritual. Now, fortunately, even before the white missionaries came to the Fore tribe, the mothers of the Fore tribe realized that their children were getting Kuru, and so they gave up this funeral ritual and refused to allow their children to eat their dead relatives as well. So when the missionaries came to Papua New Guinea, only the men had Kuru. And I think the missionaries did a service to the Fore tribe because they said, "What, you're eating their dead? That's anathema." And they stomped it out. And because the missionaries did this, the Kuru eventually became extinct in the Fore tribe. And you can argue about the missionaries being intolerant and not what are you saying, allowing the rituals of the native people. And that's all probably true. But in this case, I think the missionaries actually did the Fore tribe a service. They got rid of the Kuru. Any question about any of that? All right, there you see Kuru there. The mechanism of the disease and how it spread was there is a normal protein called PRP superscript C, which is a normal protein on the cell surface of a nerve cell, especially the brain cells, but mainly nerve cells. And it's important in the functioning of the nerve cell. And there's also a PRP superscript SC standing for the scrapese protein, which is, we're just going to call it the prion protein, which when it's in the cell, the PRP superscript SC or the prion protein can come up to the normal protein and then get the normal protein to change its folding pattern and then take on the pattern of the prion protein, meaning this protein and the normal protein have the same amino acid sequence. The only difference between them is the folding pattern of the protein. And how the prion protein replicates is it will change the normal protein into the prion protein. And then the cell will say, oops, I don't have enough of the normal PRP superscript C protein, so the cell makes more of it. But now there's two of these proteins in the cell, making it even more likely that one of them will come up to the normal protein and get it to change into the scrapese protein. The scrapese protein then will accumulate in the nerve cell, forming the plaques we find in the brain cells. And eventually, cell death will occur in this nerve cell, which causes the prion disease. You can get prion disease, like I said, from ingesting the scrapese protein, from getting in a surgical transplant, like an organ transplant, or on a surgeon's knife, which that shouldn't happen because I'm assuming they sterilize the knives beforehand. However, the scrapese protein is really resistant to destruction. We'll talk a little bit about that. And the normal sterilization process will not destroy the scrapese protein. So you could get trace amounts on a surgeon's knife. And then there's some people who are born with a gene which itself changes spontaneously from this gene to that gene. We'll talk a little bit about that. Any question about that? All right. Actually, I'm not going to talk more about that. So let me just mention there are some human families that have family members tending to get Creutzfeldt-Jakob disease or some other prion disease, and it's because they have a genetic variant of the normal protein, which spontaneously changes into the prion protein. And usually this change doesn't happen until the individual is at least middle-aged, if not older. And that means that the young adults are healthy and then they have children, passing on the normal gene, which then changes to the prion gene when the adult is older and already has children, so that it tends to run in certain families. Any question about any of that? There was an epidemic in Great Britain of mad cow disease because British, not farmers, but British ranchers, I guess you call it, wanted to use the cow brains. And so they came up with grinding the cow brains, adding it to cattle feed, meaning grain, and then they fed the grain to the cattle. So the cows were eating essentially cow brains. And one of the cows got mad cow disease. And then this practice spread it to other cows, which they then ground up the brains and gave it to the grain and then fed it to the cows. And it quickly became an epidemic of mad cow disease among the British cattle. Unfortunately, 150 British cats, both domesticated cats, meaning kitty cats, and then lions and tigers and zoological cats, came down with prion disease during this time, as well as about 150 British citizens came down with prion disease. And it was believed, in most cases, they could uh, trace it back to the patient eating tainted beef, meaning prion infected beef. Any questions about any of that? How the British finally got rid of the epidemic of mad cow disease, and I guess you'd call it Creutzfeldt-Jakob disease, is they finally incinerated all of their cattle and then imported new cattle from other countries. Any question about any of that? Uh, Let me state to kill this prion protein, for some reason, you need to get it to 1,000 degrees. I don't remember if that's Fahrenheit or Celsius, doesn't matter. It's 1,000 degrees, much hotter than any protein normally can withstand. All right, let's talk a little bit about viroids. and plant viruses. Plant viruses resemble animal viruses in many ways. They do cause many diseases of plants, including economically important crops such as beans, corn, sugarcane, and potatoes. But because of the tough cell wall of plant cells, the plant viruses tend to enter the plants through wounds or a parasite like a nematode, a fungus, or an insect, which essentially damages the plant and then makes the plant susceptible to getting the plant virus. And then the plant virus can spread either by getting in pollen or seeds and then spreading that way. We're not going to talk about plant viruses any further because plant viruses do not infect animal cells and they do not cause animal diseases. Any question about any of that? All right, and our last slide is viroids. Viroids are naked RNA molecules that have no protein coat. So you can think of them as an RNA virus lacking a protein coat. And when these RNA molecules get inside a cell, they can infect the cell and then cause the cell to make more viroids, meaning an RNA molecule, which can then go on and infect another cell, causing disease. Fortunately, of viroids, they're only known as a plant pathogen. So there are no viroids known that cause animal disease. Let me get over here. And there you can see a normal potato and then a viroid infected potato plant. How they have I don't know, a skinnier and longer potato. Any questions about any of that? So viroids do not cause human disease. We're not going to talk about them further. They only cause plant diseases like this potato spindler tuber viroid. Any question about any of that? All right, that's it for chapter 13. Let's move on to chapter 7. As is normal, I have a slide showing you the major goals of chapter 7, the control of microbial growth, and a rough outline. This is a chapter that most students have some fair understanding of anyways, because, well, you know about antibiotics and disinfectants and antiseptics, and these are agents which control microbial growth. And you know about sterilization and disinfection and antisepsis, degerming, sanitization, autoclaving, pasteurization, and maybe not biocide and germicide, but we'll talk a little bit about those. Then number 2, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways in which an antimicrobial agent kill or inhibit cells. And then 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance the major microbial groups have to antimicrobial agents. Any question about that overview and rough outline? Let me blow this slide up. So when we're talking about the microbial death rate, a bacterial population dies at a constant logarithmic rate when it's heated or treated with antimicrobial chemicals. Meaning if you have 1 million survivors at time 0 and then expose them to an antimicrobial agent, after 1 minute, this is a hypothetical example, you'll have about 100,000 survivors, meaning only 1/10 the population has survived, and there will be 900,000 deaths. And then you expose it to 2 minutes, you'll have only 10,000 survivors. Expose it to 3 minutes, you'll only have 1,000 survivors. Expose it to 4 minutes, 100 survivors. Expose it to 5 minutes, 10 survivors. Expose it to 6 minutes, and you'll have only one survivor out of 1 million. The constant logarithmic death rate is, meaning on a log scale and time, you get a linear death rate like that, which is why microbiologists like using a log scale, because the death rate is logarithmic, and then you have a very easy equation, y equals mx plus b. You could plot it on a arithmetic scale, the number of survivors, and the x-axis is always arithmetic scale, and then it's a death rate, logarithmic death rate, which is much more difficult to explain arithmetically. Any question about any of that? The point is, when anything is exposed to when a microbe is exposed to an antimicrobial agent, it has a logarithmic death rate where the death happens logarithmically in a straight line, something like this. Any question about any of that? The rate of microbial death can be affected by a number of different variables. So the effectiveness of the antimicrobial treatment will depend on the number of microbes present at the beginning of the treatment. The more microbes you have present at the beginning, the longer you need to treat the population to reduce the numbers to safe levels. Like if you wanted zero, which you would in something like Ebola virus or anthrax, you need a very long exposure time to make sure you have zero. But on most microbes like E. coli, you just need to decrease the numbers to safe levels, like 10, maybe even 100 would be safe. Even in food, eating 100 E. coli, normal E. coli would be safe. There are environmental influences that can affect the rate of microbial death. For example, the presence of organic matter, which is almost always seen in hospital specimens, can influence the death rate. And a good example is bleach can become inactivated by organic matter, like in fecal matter or even dandruff. You have organic matter around, and so you need more bleach to kill off all of the microbes, as well as perhaps a longer time because of the organic matter. The temperature of the treatment can affect the environmental influence. Generally speaking, the warmer the temperature, the more effective the microbial death rate will be. That depends a little bit on what you're talking about and what temperature you're talking about. But generally, when you heat the temperature, the agent will kill the microbe sooner. And then the presence of biofilm can affect the number of microbes' death. Generally, a microbe that has no biofilm will die much easier than the same microbe which has a biofilm. Any question about any of that? The time of exposure, of course, affects the microbial death rate. You need longer time for more cells. You need longer time of exposure if you have endospores. Microbial characteristics can affect the microbial death rate. I've already mentioned one, and that is if the population of bacteria has endospores, you need a much longer time of exposure than you would with the same bacteria species that had no endospores. We'll talk more about microbial characteristics affecting the microbial death rate near the end of this lesson. Any questions about any of that? All right, here's some terminology you should know. Um, I'm only going to talk about some of these. Sterilization. That's the destruction of all um, microbial life, with the possible exception of prions, because prions are really difficult to kill. Most things, most disinfectants do not perform sterilization. They can disinfect, but they usually do not kill all endospores. You can kill all things using an autoclave, except for prions, of course, and maybe sterilizing gas, such as ethylene oxide. And then there's one other one, hydrogen peroxide or the other peroxide agents can kill all microbes, meaning they do kill endospores. But most disinfectants do not kill endospores, in which case it doesn't sterilize. All it does is disinfect. Disinfection refers to the destruction of vegetative pathogens, not necessarily endospores. And its disinfection is on an object. If it's on a living tissue, we call it antisepsis, destruction of vegetative pathogens on living tissue. So if you use bleach to kill the microbes on a toilet and you really only disinfect, you're not killing the endospores on the toilet. We call it disinfection. But if you were to use bleach, such as to treat somebody who has necrotizing fasciitis, also called flesh-eating bacteria, that would be called antisepsis, using the bleach on living tissue. Degerming is the removal of microbes from a limited area, such as the skin around an injection site. So when we alcohol swab where we're going to inject, it's actually only degerming. It does not kill all microbes. All it does is reduce their number, making it safe to do the injection. Sanitization is a term used using high temperature washing or dipping in a chemical disinfectant to lower the microbes to safe counts for eating. This is usually done in public health levels, like at restaurants, to reduce the microbes to safe public health levels. Any question about any of that? All right. A biocide and germicide are substances that kill microbes. They do not usually kill endospores. Like alcohol can be considered a biocide or a germicide, and it does not kill endospores. A bactericide is something that kills bacteria. A fungicide, something that kills fungus. Viricide, something that inactivates viruses. Bacteriostasis will inhibit the growth of the microbe, but when we remove the bacteriostatic agent, growth might resume because the agents were only inhibited, they were not killed. I actually know of no disinfectant that acts like a bacteriostatic agent, but my understanding is there's some antibiotics which act as a bacteriostatic agent. And then we use these bacteriostatic antibiotics with the understanding that the host immune system will remove the agent from the patient so that when you stop giving the bacteriostatic agent, the disease doesn't come back. Any question about any of that? All right. Sepsis refers to microbial contamination. Asepsis, the absence of contamination from unwanted organisms. Like if you were to come into the lab, I would hopefully give you aseptic media, which has no contamination in it, and so nothing should grow in it until you inoculate the media with whatever you want to grow. You've learned aseptic techniques, obviously online, but they are important in the hospital setting and especially surgery to minimize contamination from the environment, such as the air the surgeons blade when surgery goes on. Any question about any of that? All right. Antimicrobial agents kill or inhibit microbes by one of three means. You can alter the membrane, damage the membrane, alter membrane permeability. You can damage intracellular proteins, and then you can damage nucleic acids. These are the three ways antimicrobial agents kill or inhibit microbes. Polymyxin B damages the cell membrane or another membrane, damaging the membrane, causing the cellular contents to leak out of the cell, killing the cell. Uh, Alcohol can damage the cell's proteins or the microbes' proteins, such as inactivating the enzymes. Alcohol can also damage the membrane permeability, by the way. You can damage the cell membrane with alcohol. So, alcohol, my point is an agent that can damage cellular proteins. Another agent that can damage cellular protein is something like proteinases. We don't usually use proteinases to use as an antimicrobial agent, but you could because they damage proteins, which would be essential to the microbe. And then the third way is you can damage nucleic acids. I think you're all aware that radiation, both UV light and X-rays and gamma rays, can damage the nucleic acid, which you get enough mutations in the cell, you need something like 5 or 6, you can kill the microbe. So you damage the nucleic acid, you cause mutations, you get enough mutations, it will kill the cell. In fact, if you get a mutation in a critical gene, you'll kill the cell with just one mutation. Any questions about any of that? All right. There are different physical methods to control microbes. You can use heat, filtration, low temperature, high pressure, desiccation, osmotic pressure, and radiation. All of these are physical methods we have to control microbial growth. Heat, all methods kill microorganisms by denaturing proteins and enzymes, though very high heat, like inflaming and incineration, turn the cells into char. There are two types of heat, wet heat and dry heat. Boiling is an example of wet heat. Dry heat, an example of that would be baking. Obviously, there are other examples like wet heat can also be autoclaving and steaming, which is usually much higher temperature than boiling. And then dry heat can be much higher temperature than baking something in your oven. You can flame it, pass it in the flame, or put it in the back to incinerator, incineration, much higher temperature. Baking will only kill the microbe by denaturing proteins, but like I said, flaming or even incineration will turn the cells into char. Any question about any of that? Okay, moist heat denatures proteins and enzymes, killing the cell. Boiling is only at 100 degrees Celsius or flowing steam. Kills vegetative pathogens, most viruses, fungi, fungal spores, within about 10 minutes of exposure, often much faster. Like you simply bring the solution up to 100 degrees, meaning get it to boil, you kill all E. coli because it dies much easier than most, well, not most microbes, but many other microbes. And you don't need 10 minutes of exposure to 100 degrees C to kill something like E. coli. Endospores and some viruses take considerably longer to destroy. Some endospores can last easily much longer than 10 minutes, can take over 20 hours of boiling water temperature to kill the endospore. So how do we get reliable sterilization? By moist heat, we bring the temperature above boiling, like in an autoclave, where you have steam under pressure. And so it's more than just steam in the autoclave, meaning it's usually 121 degrees Celsius at 15 pounds per square inch pressure. 1 pound per square inch, I think, is atmospheric pressure. Is that right? Can somebody confirm that? Let me go look on that. There it is. Actually, that's 14.7 pounds per square inch. I'm glad I looked that up. So 15 pounds per square inch is just a little bit above atmospheric pressure. Excuse me here. Ah, and that's probably because when you have atmospheric pressure higher than atmospheric pressure, you have to contain that in a closed environment to keep it safe. But anyways, the autoclave puts the steam under pressure, being much more likely to kill the microbes. In fact, in 1 liter of something autoclave for 15 minutes at 15 pounds per square inch, you get killing of even all endospores. So that in 1 liter, you'll kill all endospores within that 1 liter. Any question about that? And you only need 15 minutes of exposure time. And that's how we get things sterilized in the microbiology lab, usually with the autoclave, which is simply, if you don't know, an autoclave is like a pressure cooker. Works similar to a pressure cooker. In fact, all it is a giant pressure cooker. All right, let's talk a little bit about pasteurization. Pasteurization reduces the number of spoilage organism and kills all pathogens, but it does not kill all organisms. It is mild heating sufficient to kill organisms that cause spoilage and tends to kill all pathogens. But the thermoduric organisms, these are heat-resistant organisms, can survive pasteurization. And that's why pasteurized milk does have a shelf life, because it does have microbes in it, which can eventually spoil the milk. So the milk that is pasteurized has a longer shelf life than unpasteurized milk, because unpasteurized milk has all the microbes in it. And pasteurized milk has reduced numbers, but still has microbes in it. Pasteurization, there's two pasteurization techniques. 63 degrees, bring the milk up to 63 degrees Celsius for 30 minutes. Most commercial milk is brought to 72 degrees Celsius for 15 seconds. And most pasteurization of everything, like beer and milk, even antibiotics or something like that, is brought to this 72 degrees Celsius for 15 seconds. But you could expose it to 63 degrees Celsius for 30 minutes. You can have ultra high temperature milk, UHT milk, which is actually sterilized. So this is not pasteurized milk. And you have to bring the milk to 140 degrees Celsius. That's over boiling temperature. Boiling, remember, is 100 degrees Celsius. You only need to bring it to 140 degrees Celsius for 1 second. And then you can sterilize the milk. And then the milk can be cartoned in something that prevents growth from getting at it and can be stored at shelf life. Does not need refrigeration till it's open. Any question about any of that? All right, this picture is showing you that baking is not as efficient at killing microbes or this cat uh, from dry heat versus wet heat. And you actually, guys, you all know this because all of us stick our hands in an oven to, in this case, pull out the cat or pull out what we baked. And you can put your hands in the oven for a short period of time. However, you cannot put your hand through steam. Even if the steam is only 100 degrees Celsius, you cannot put your hand through steam or in boiling water for any temperature without getting burned. But baking, let's say 350 degrees, you can put your hand in there. And this cat climbed in a warm oven, which actually was hot because it liked the warmth. Obviously, this wasn't so hot, the cat was going to get burned. But the cat is showing you that warm heat or dry heat is not as efficient at transferring energy as wet heat. Any question about any of that? All right. Dry heat kills by oxidation, and obviously flaming and incineration changes the cells into char. So an oven would be something like 350 degrees or 170 degrees Celsius for 2 hours is needed to sterilize the environment, something we bake. And you don't need that long at a much cooler temperature, 100 degrees Celsius, when we boil things. You only need about 10 minutes to kill most things in boiling. Any question about that? All right. Another physical method we have for controlling microbial growth is filtration. Filtration removes microbes from liquids and gases when we pass it through a filter. You obviously can't pass a solid through a filter, but we could put a liquid or a gas through something that has a pore, trapping the microbes in the pore, and then what comes down would be filtered and could be sterile. Examples of some things that are sterilized by filtration include some juices. Coors beer, if you're familiar with beer, is actually filtered. It is not pasteurized. And this is something I know because Coors beers was not allowed to be sold in Oregon because the state law was you had to pasteurize the beer before you could sell it. And Coors eventually got an exception because they sterilized their beer by filtration, which supposedly makes the beer taste different than pasteurized beer. Certain vaccines, milk is usually not filtrated because the fats and the proteins in milk will clog the pores in the filter, and then you can't filter the milk. So milk is usually not filtered. Any question about any of that? All right, low temperature can be used to control microbial growth. Low temperature inhibits microbial growth. It may not kill microbes. In fact, most microbes, not all of them at least, would be killed by low temperature. I guess one exception would be a eukaryote microbe. A eukaryote microbe would be killed by freezing. It probably would not be killed by refrigeration, but refrigeration will slow the metabolic rate of the microbes. It cannot reduce the microbe can't reproduce at refrigeration temperature, or at least many microbes cannot. Obviously, some microbes can, and it would prevent the human pathogens that make toxins from making toxins as well. But the psychotrophs can reproduce at refrigeration temperature, and they will reach about 2 million cells from one cell in about a week. And that's why food, when you put it in the refrigerator for a week, it should be eaten within a week or thrown out because at 1 week, the psychotrophs can grow to numbers to spoil the food. Freezing can stop antimicrobial growth. You freeze the water like your home freezer. It freezes the water, forming ice crystals. The ice crystals can, how do I say that? Can kill some bacteria, but usually not all bacteria. Even when you're talking about one species, freezing and then thawing tends to only kill about 50% of bacteria. It does not kill any endospores, freezing, and there are some species which are resistant to freezing. But freezing does kill all parasitic or eukaryotic microbes. So, like a worm, you freeze the pork, for example, you're going to kill the worm eggs in the meat. And the same with yeast. You freeze it, parasitic yeast, you freeze it, it'll die. But bacteria can, at least about 50% of them, can survive freezing and thawing. Quick freezing with liquid nitrogen does not kill bacteria cells, but slow freezing, which is in your home freezer, can kill some, but usually only about 50% of a species, and then the other 50% can survive. And it's because the ice crystals just happen to kill the bacteria, the one that dies, and then the one that doesn't have a lethal ice crystal in it. It might have an ice crystal in it, but it's not in a lethal position in the cell. All right, any question about low heat? Low heat, like I said, for bacteria, the main thing is it just stops the growth. Freezing totally stops the growth. Low temperature, well, actually, that does inhibit most microbes, but obviously the psychotrophs can grow at refrigeration temperature. High pressure transfers pressure evenly throughout a liquid, killing all vegetative bacterial cells. Does not kill endospores, but it can be used to make certain liquids safe. It denatures proteins, alters carbohydrates. Endospores are resistant to high pressure, but you can use high pressure to preserve the color, the flavor, and the nutrient value of juices. And heating usually changes both the color, the flavor, and the nutrient value. Like if you don't know, vitamin C is destroyed by heating. And if you treat the juice with pressure, you can retain the vitamin C as well as the original color and the flavor. There's a little joke here. We have the junior Cole and the mother Cole telling, Dad's been under a lot of stress lately. Carbon and coal has been changed to a diamond under high pressure. Desiccation is drying microbes. It prevents all microbial metabolism. Some cells can remain viable for years, though, like cells that have endospores. But while the cell is desiccated or dried out, the cells cannot grow. Resistance to desiccation varies by species. For example, E. coli cannot survive desiccation very long. At most a day, probably less. And that's why when E. coli falls into molasses, the water will be sucked out of the E. coli by the molasses, desiccating the E. coli, and that would kill the E. coli. However, an endospore can survive for, well, decades in molasses. It can't grow, but it could survive. Osmotic pressure is using high salt, high sugar to cause plasmolysis, where the plasma membrane will pull away from the cell wall. That can kill many cells. And we use either osmotic pressure, high salt, or high sugar to preserve some foods, as well as drying out the food. So examples of desiccation include dried fruit, dried figs. Examples of osmotic pressure, molasses and honey. The high osmotic pressure prevents these food from spoiling. And as I mentioned, my mother had a bottle of molasses that was good for at least 20 years because it never killed my father, and probably for 30 years before she ever threw it away. Some other physical methods that can control microbial growth include radiation. Radiation damages DNA, nucleic acids. Ionizing radiation has high energy, short wavelength, high energy. This ionizes molecules in water, which produce ions, which can then go on and react with other organic molecules, such as DNA, causing mutations and damaging the DNA, killing the cells. Excuse me here. Radiation, particularly ionizing radiation, is used to sterilize food, pharmaceuticals, and medical supplies. You might have seen some camping food in the store, and it's closed at room temperature, and it's going to stay good because it's been ionized, exposed to radiation, and so it's sterile from DNA damage. Let me remind you that uh, we have ionizing radiation that has a short wavelength, a lot of energy. When it hits a electron, it causes the electron to leave the atom, creating an ion, which can damage DNA. With UV light, it can damage DNA by making two adjacent thymines, forming a dimer between them, damaging the DNA by what we call a thymine dimer, inhibiting DNA replication as well as RNA transcription. Microwave radiation is not particularly antimicrobial, but it can kill microbes by causing the microbe to heat up. All right, there's a summary of some of the different methods of antimicrobial, physical methods of antimicrobial growth. You should just really know the terms. Let's move on to the chemical methods of microbial growth. There are few agents that achieve sterility. Most simply decrease the microbe numbers to safe levels or remove vegetative forms of the pathogen from an object. Like when we disinfect a toilet, you don't remove the endospores with bleach. You do remove all of the vegetative bacterial forms, though, with bleach. So disinfection refers to the destruction of vegetative pathogens. The different factors related to effective disinfection are the concentration of the disinfectant, the time. Obviously, the disinfectant has to be able to access the microbe too, like UV radiation only gets the surface. And if the microbe is below the surface, you can't disinfect it with UV. UV is not a chemical disinfectant, by the way. It's just an example I'm talking about here, where the surface, your disinfectant has to get deeper than the surface if the microbe is deeper than the surface. The pH can affect the disinfection. Generally, the more acidic the pH, the better the disinfection. And it's probably true that the disinfectant will be least effective at neutral pH. The presence of organic matter can affect the disinfection, Organic matter is usually found in clinical situations, like in a hospital. An example of this would be bleach can be inactivated by organic matter. There are different types of disinfectants, phenol and phenolics. Phenol was the strongest disinfectant that, oh, what was that guy's name? Drawing a blank here. The guy who started aseptic surgery. Phenol was the strongest agent he found to be a disinfectant, and so he treated his patients before surgery with phenol to get it to act like a disinfectant. This actually is irritating to the skin, phenol. It's not something you really want to breathe in. And then he didn't know this, but phenol is actually a carcinogen as well. Today we have phenolics, biphenyls, which are better. And then we have biogonides, halogens. Let me mention two halogens. Bleach is a halogen, chlorine bleach. And then tincture of iodine, another halogen. Alcohols, it's actually a weak disinfectant. Generally, we call it a degerming agent because alcohol is not a very good disinfectant. We usually do not clean a toilet with alcohol because it does not kill a lot of microbes. Heavy metals, its use has come back as a disinfectant, like in surgical dressing. We can put heavy metals on the dressing to prevent a bacterial infection. And heavy metals has come back because of the appearance of drug-resistant variants. And usually the drug-resistant variants will be sensitive to the heavy metal, and then the heavy metal does not encourage the formation of drug-resistant variants. We have surface active agents like soap and disinfectants, and there's surface active agents used in the hospital now. Chemical food preservatives, there are antibiotics and aldehydes. Gaseous chemosterilizers, a very effective chemical disinfectants. This is one of the ones that can kill endospores, but you need long-term exposure to kill endospores. And then peroxygens, these are oxidizing agents like hydrogen peroxide, another very effective chemical disinfectant that can kill endospores. So these last two are the main ones that kill endospores, a disinfectant that can kill endospores. Here are the terms you should know. I'm not going to go through them. You only really need to know the term. I'm running out of time. That's why I'm skipping going through these terms, but you should know the terms. Comparing the effectiveness of various antiseptics. Soap and water, I remember we had this in a lab, is not a very good disinfectant. As you can see, washing your hands with soap and water will only kill about 10% of the organisms after 120 seconds washing your hands. And I don't know about you, but I never wash my hands for 120 seconds. I wash my hands for at most 30 seconds, like when I'm in the lab. You're supposed to wash your hand for about 30 seconds. And that's killing less than 10%. So soap and water is not a very good antiseptic. A good antiseptic that is found in most homes is actually tincture of iodine, which most people just call iodine, killing more than 90% of the organisms at 120 seconds, actually more than 90% at 100 seconds. Tincture of iodine used to be used before surgery, meaning they would treat the patient's skin with it, and then the iodine would stay on the skin taking a long time to sterilize the skin. And then the advantage was the surgeon could see where the skin was treated with iodine and make sure he doesn't cut outside that region treated. We have better agents now than tincture of iodine, which tend to be used in the hospital setting. All right, any question about any of that? Some of you could tell me what some of those agents are they use in the hospital. I'm sorry, I'm not familiar with what they use in the hospital anymore because I'm not in the hospitals. Should look it up. Nobody have a suggestion? All right. Um, let's talk about some of the microbial characteristics that can control microbial growth. And we're talking about all microbes in general. The most resistant of all microbes is prions. Very few things kill prion. You need 1,000 degrees Fahrenheit to kill a prion. Endospores, the next most resistant microbe, Mycobacterium, all that mycolic acid in the cell wall prevents any water-soluble agent from getting inside the cell and then killing the cell. So Mycobacteria, the third most resistant. The cystiprotozoa, the next most resistant. Vegetative protozoa, gram-negative bacteria tend to be more resistant than gram-positive bacteria because of that outer cell wall, preventing certain agents from getting at the cell and then killing the cell. Fungi, the next. Viruses without envelopes, the next. Gram-positive bacteria, the next. And then the least resistant, viruses with lipid envelopes, like COVID-19. Anything that damages the DNA, the protein of the virus, will damage the virus, as well as anything that will damage the lipid envelope can damage the virus. Why they're so susceptible to damage, as I mentioned, with coronavirus, you only need a day at room temperature to dry out the virus and then kill it if it's on paper or cardboard. If it's on plastic or metal, you may require 3 days at room temperature before the virus will die. This is a simplified hierarchy showing you the relative resistance of the major microbial group to most chemical biocides. Please understand that there are factors that come into play here that can differ from what I've shown you. Let's talk a little bit about the gram-negative bacteria having their outer cell, outer wall of the membrane. If the agent can get through the porins, the agent could get in and kill the gram-negative cell. But if it can't get in the porin, then the agent cannot get in the gram-negative bacteria cell and then damage it. One example of an agent that cannot get in the gram-negative porin is an antibiotic. Sorry, I'm drawing a blank here on this name. Penicillin G cannot get in the porin of a gram-negative cell. So penicillin G does not kill gram-negative cells, but it can get in the gram-positive bacteria and then damage the cell wall. I mentioned the mycobacteria are resistant to any agent that is water soluble, like bleach. Can't get in the mycobacteria and damage the mycobacteria. So mycobacteria are fairly resistant to the most antimicrobial agents. However, mycobacteria are very sensitive to alcohol. As you remember, I did mention that alcohol was not a very effective disinfectant because it wasn't very good at killing cells. but alcohol can get right through the mycolic acid. And then the mycobacteria are very sensitive to treatment with alcohol. So the mycobacteria, which are very resistant to most disinfectants, are, because of their characteristic, very sensitive to alcohol. Any question about any of that? And like I mentioned, endospores are resistant to most disinfectants. The exception would be the gaseous sterilizing agent, which you need at least 20 minutes exposure to kill the endospore, or something like hydrogen peroxide. The peroxygens can kill the endospores very easily. So you only need limited exposure to hydrogen peroxide, then you can kill the endospores. Any question about any of that? All right, let's talk about prions. Prions are the infectious protein agents that cause spongiform encephalopathy. How do we kill them? It's not completely solved yet. You can, like the British, incinerated their cattle at high temperature, 1,000 degrees. I don't remember if it's 1,000 degrees Fahrenheit or 1,000 degrees Celsius. It's really high. You can kill the prion. You can't obviously treat most things to 1,000 degrees and get it to survive. Like glass can't take that temperature. Surgical instruments can't take that temperature. So how do we treat surgical instruments? Well, they found that if you treat the prion to a proteinase, the proteinase can digest the prion and then remove the prion from the surgery instruments. So we do have a few things, but most agents do not kill prions. Any question about any of that? All right, final comments. Microbial growth methods are effective, especially the biocides, but they're not uniformly effective against all microbes. The compounds discussed in this lecture are generally not useful in treating a patient with disease, except for the antibiotics, which I just mentioned only in passing. Meaning you cannot treat a patient with bleach to remove the microbes from the patient. If you remember once, Trump suggested maybe we should treat people with bleach to cure them of COVID-19. It does not work. You can't treat a patient with bleach. That will kill the patient. Any question about any of that? All right, let me give a brief review of this lesson because it's not going to be in the review that I'm going to post. Because I only talk about chapter 7 in the fall. You should know the terms of this chapter. Actually, you should know the objectives more than just the terms. You should know the factors that influence microbial death rate. The number of microbes present, you need longer time. If there's endospores, you need longer time if the agent even kills endospores. The presence of organic matter can influence the amount of cells that are dead, like in a hospital specimen, like a fecal accident. You need lots of bleach if that's what you're using to kill the microbes, because bleach is inactivated by organic matter. Inorganic matter, I mean. Yeah, no, organic matter. The temperature can affect the treatment, the presence of biofilm, like treating an earache or dental plaque has biofilms. You need longer term treatment. Guess I never thought about that when you're using something to kill the microbes in your mouth, like mouthwash, if there's biofilm there, you're probably not going to kill the bacteria cells. However, you can use hydrogen peroxide as a mouthwash. You have to reduce the concentration. I think it's 50-50 with water. That would probably kill the microbes that has a biofilm in your mouth. Turn that off. Somebody's calling me. Um, and then the microbial characteristics can affect the number of the death rate. Whether the cell has myco is part of the mycobacterium family, has mycolic acid in the cell wall, or whether the cell has endospores, you'll need much longer exposure if the agent can kill the microbes. Let's go on. You should know a biocide, a germicide, bactericide, fungicide, virucide. Biocide kills the agent. A bacteriostatic agent just inhibits the growth of the agent. When you remove the bacteriostatic agent, if the microbes are still left, assuming they're still alive, they will continue to grow. We had a lab about that too. All right, you should know the three modes of killing microbes: alter the membrane permeability, damage the membrane, damage to intracellular proteins, and then damage to nucleic acid. These are the three ways that all antimicrobial agents kill microbes or inactivate them. Well, on this one, you should know, well, you should know the different physical methods and chemical methods of controlling microbes. You should know that wet heat is more efficient than dry heat at killing microbes because wet heat transfers the energy in the heat to the microbe much quicker. You should know that low heat does not necessarily kill the microbe. Even if you freeze, most bacteria can survive. Freezing, at least slow freezing, does kill all eukaryote pathogens, but not bacterial pathogens. You should know that radiation damages cells by damaging the DNA. Two types of damage: ionizing radiation, creating ions, and then UV radiation, creating thymine dimers. Both damage the DNA. And then the chemical agents for controlling microbes. Um, there we got the different chemical agents. The physical agents, which I already showed you, are things like radiation, heat. Chemical agents are a chemical that we use to mainly control the microbes, like phenol, bleach, a halogen, alcohol, like rubbing alcohol, heavy metals. What's a common one? Silver, copper can be used to prevent microbes growth. There are certain antibiotics that we put in cheese, for example, and that prevents microbes from growing in the cheese. I didn't mention that. You don't really need to know that. I think niacin is the antibiotic. Let me see if I can find that. Chemical food preservatives. Nope, it's not antibiotics there. All right, and then know the microbial characteristics that can affect the death rate. All right, it's about chapter 7. Any questions about this class? All right, if there's no questions, let me, wait a minute, share my screen because I didn't talk about the final. Let me mention that the final will be due. Come on, get out of the way. Oh, that's odd. I don't have it here. Maybe it's on the last page there. There it is. The final will be Wednesday, December 11th. You must get it done by 11:59 p.m. online. There will be heavy coverage of the chapters that you have not been quizzed on, meaning chapter 7, not quite so heavy on that one because we only did it today. Chapter 13, chapter 14, and then lab module 9, as well as it isn't on here, but there will be a few questions on the unknown project. We're dealing with the unknown project. And then that'll be about 50% of the questions on this material here. 50% of the other questions will be on the previous material that you have been quizzed on, meaning labs 1 to 8 and chapters 1 through chapter 8. Let me do mention that I do like the history of microbiology, and chapter 1 also introduced a whole lot of concepts to you that we then later went on and talked about. And so there will be a few more questions on chapter 1 than on the other chapters. As far as chapter 2 goes, I think I've stated this in the review. Other than you should know what the chemical is, I'm not going to ask any questions directly on chapter 2. Meaning I might ask a question and expect you to know what the chemical is, but I'm not directly asking a question about chapter 2. Any questions about that? Any questions about the final?
I do have a question as I was looking through the grade book. It says that there's a final B in parentheses that has everyone take it, and then a final A, then it says all other students for 15 points. You want us to take both of those?
The final A is fill in the blank or essay questions. There's only a few questions in final A. One student is going to be taking final AA. Okay, so that's why I said most students will be taking final A. And then everyone takes final B. That's the multiple choice questions. Most of the questions are on final B. Good question. I forgot to mention that.
Thank you.
Okay. Anything else? All right. It's been good teaching you. Good luck on your final. Good luck in your career. And I hope you do well. Bye.
Thank you. Oh, did you say that you were going to be in lab to go over a review, or was that what we just did?
No, I only reviewed chapter 7 because review I'm going to post will not have chapter 7 in it. But I do have a review that I'm going to post. I'll put it in the lab slot, but there will be a review that you can go over if you wish during the lab time or whenever you want. I will
Gotcha.
review. Okay.
Yeah, thank you
Okay.
so much.
All right. Bye.